Okay, next we will hear from our last presenter, Dr. Ken Sims, PhD, Senior Materials Scientist at Battelle.
Right. Thank you very much for that kind introduction. And thank you to the programming committee and the meeting organizers for this opportunity to share this work with all of you today. Especially thank you to Kate and Vanessa for coordinating logistics and all that fun stuff. So just jump right in. These are my disclosures. I work at Battelle. If you're not familiar with Battelle, Battelle is the world's largest independent nonprofit research and development organization. We're headquartered in Columbus, Ohio. Our mission is to translate scientific discovery into societal benefits. So we literally live in that valley of death between basic research and practical application. In the past, we helped launch Xerox, the UPC symbols that you use at the grocery store, and lots of other technologies. And we've recently, in the past several years, gotten involved in gene therapy. Our profit serves a purpose in that we reinvest into STEM education and help manage different schools within the Columbus Metropolitan School District. And we also manage 9 national laboratories for the federal government. So we also operate as a federal contractor. We operate in the space of national security, environment, infrastructure, and health. And obviously, it's the latter part that I'm here speaking about today. So we're working with the Gilbert Family Foundation on a project focused on neurofibromatosis type 1. I probably don't need to belabor this point to this crew, but basically, if you're not familiar with NF1, it's basically mutations in the NF1 tumor suppressor gene. This dysregulates the RAS-MAPK pathway, resulting in brain and nerve sheath tumors. So as we've already heard several times today, obviously delivery is the main problem, particularly to the brain and to the nervous system. So our project is really focused on delivering to the brain and the sciatic nerves, particularly the Schwann cells for neurofibromatosis. So working under some funding from the Gilbert Family Foundation, we're working with a colleague at UC Davis, Dave Segal, and another colleague, Kyle Fink there. And Dave Segal's lab kind of took on our first aim of our project, which is really to develop the payload, in this case, a plasmid that would encode for these epigenetic regulatory proteins, or ERPs, that would help regulate the MEK pathway, or basically regulate MEK 1 and 2 in the RAS pathway. So they started this project from their end, looking at the payload. They initially designed 20 different guide RNAs for this CRISPR plasmid, created the guides, tested their repression capabilities, and ultimately landed on 2, one focused on MAP2K1 and one focused on MAP2K2, and demonstrated significant repression of 70 and 95% respectively across those. And they were able to show in neural 2A cells that these constructs could actually maintain suppression for over 3 weeks, or up to 3 weeks. So this was a good start. However, these are 2 separate plasmids. So we really needed to get both of these entities combined into a single construct so we could make the delivery a little bit easier. So that grad student handed off the project to a postdoc who then was able to put both of these constructs into a single plasmid using a CMV promoter. We did see that the MAP2K2 repression levels were maintained. However, the MAP2K1 were not. After lots of work, they've really kind of leveled out at about a 40 to 50% repression level. However, the particular plasmid here, they were able to get both of these constructs into a single plasmid a while back, and then were able to hand that off to our team at Battelle so we could actually test them with our delivery vehicles. So moving on to the second aim of our project, which is really where Battelle picks up using a platform that we have focused on polymer nanoparticles to deliver these plasmid DNAs to Schwann cells. Our platform is called HitScan. So this is an acronym that stands for High Throughput Synthesis, Characterization, and Assessment of Nanoparticles. It's really built on 3 different pillar technologies. First is automated polymer library synthesis. Second is high throughput in vitro and in vivo screening. And then third is combining the data from the first two using a machine learning algorithm so that we can drive this design, build, test, learn flywheel and basically iterate rapidly to hopefully identify final constructs that were better than what we started with. And so for our polymers, we're using a raft polymer process. I won't belabor the chemistry or any of the points on this, but happy to talk afterwards if anybody's curious. But for this project, we're basically staying right here in this kind of linear copolymer range. But we do have capabilities to do some of the others and have leveraged those on other projects. But for when I'm talking about a nanoparticle, for now, if you need a visual, you can kind of look at the self-assembled micelle at the bottom of the slide. So after we make our nanoparticles, then we actually also have in-house capabilities to track them and test for biodistribution and functional performance assays, basically. So we can use a variety of different approaches. So we have a PMP tracking or barcoding technology. We also can use fluorescence to track either the payload or the nanoparticle, sometimes both. We also can look at the different cargo and look for the expression of a fluorescent cargo. From an early preliminary screen, we had actually identified proof of concept using our DNA barcoding technology with the nanoparticles that we could find different PMPs that seemed to go to more prevalently to different tissues, in this case, the heart and the lung shown there. So by incorporating these different technologies, we can screen rather quickly through different polymer constructs to look for one that is best suited for our application. In this platform, we basically use a process where we can first we start off with some early exploration. We can identify these PMPs with specific quality attributes. We can converge rapidly on that design space, really hone in on increasing our specificity and performance, and then diverge as needed for different outputs that we may encounter along the way in all of our testing. And we've actually done this for this Gilbert Family Foundation funded project. We had started a number of years ago where we actually had some preliminary data from an earlier screen where we had a library of nanoparticles that seemed to distribute pretty ubiquitously throughout the mouse. So that seemed like a good place to start. Now that was administered through IV. And for our project, we're looking at intrathecal. So we ended up designing a library off of that. We found that in vitro, we only transfected 4 to 6% of our Schwann cells, which was not great, but it was at least an initial starting point. We did find a number of nanoparticles that were distributed to the sciatic nerve and the brain, and they were all clustered together on one of the 6, like one region of one library out of the 6 libraries that we had tested. So when we started with 1,000 different polymer designs down to 300 for this particular project. And then we iterated again for round 2, where we kind of got it down to about 50. And we were really able to focus in on this particular design space and play with that a little bit and found a few more regions of very specific interest associated with those polymer designs. So in our second round, we were able to increase our Schwann cell transfection up to about 15 to 20%. So it was a 4-fold increase, still maintaining pretty good viability. Oops, I'll go back. There we go. Pretty good viability and loading of our plasmid construct. Further enhancements during that second round, looking at our formulation. So as we started to play with different ratios of our plasmid and the nanoparticle concentrations, we're able to improve our Schwann cell transfection efficiency even further, up to 40 to 50%, which ultimately was about 10-fold higher than round 1. So we identified our top candidates here, and we're able to take those forward into some animal studies. And ultimately, when we look at the round 1 to round 2, you could see we were able to really start to dial in terms of size, polydispersity index, which is basically a measure of our variability of our nanoparticle population. The plasmid DNA loading efficiency, we're able to maintain that while increasing our Schwann cell transfection efficiency and maintaining the cell viability. So ultimately, we identified our top candidates, and we're then able to move forward into kind of a functional performance assay, which was the last aim in our study. using our top candidates. So as was mentioned earlier, manufacturing is a key consideration. It's definitely a key challenge. So the way that we've built our platform, we actually started with manufacturing in mind from the get-go so that we can perform our reactions and all of the polymer production at a scale that would scale out rather than scale up. So if you think of an assembly line, we can just make more of these small batches of polymers that would in essence go straight into a vial and then to the clinic, rather than having to play with all of the tech transfer challenges that come with scaling up some of these capacities. And so this is just a proof of concept study showing that we have been able to successfully do this with 4 different batches where each batch had 20 different replicates. And you can basically see all of the replicates look more or less the same. And then in terms of our molecular weights and our zeta potentials, overall pretty good. So this data really suggests that we can, rapidly produce a sufficient amount of materials and just scale out as needed to produce material for the next animal study, so we can accelerate our in vivo testing. So then for our top 3 nanoparticles of interest for this particular study, we kind of ran the same data set. Now we did have a deviation, so we weren't able to get some data on one of the candidates. So then we had an odd, unexpected zeta potential result in another. But by and large, size and some of the attributes were pretty closely comparable across these. So in our functional performance mouse study, we injected intrathecally to mouse on day 0 and then tested in the brain, in the sciatic nerve, in the liver on day 5. We also tested this against selumetinib, since the goal of this entire effort is to essentially try to create a once and done selumetinib like MEK inhibitor therapeutic. And I will say that we did not see any adverse events in any of the mice due to any of the control or test article doses. In this case, and we also had previously done a side study where we actually looked for, separate from this, looked for the DNA actually in the tissues of interest, and were able to see those when we just looked for the DNA. So we knew at this point that our nanoparticles were safe. We knew that they were distributing to the tissues of interest and taking our payload to the regions of interest. So we're really curious, would they actually perform as we expected? And unfortunately, in our first go round, they did not. So here you can see that we really did not see any significant differences in our MAP2K1 or MAP2K2 expression in our overall results in the brain, sciatic nerve, or the liver. We also didn't see it in our control, which was also a little bit problematic. So we're working to repeat this. However, when we kind of splice the data, looking at it from a sex standpoint, you could see a really interesting pattern emerges in the females, where we actually did see for our MEK2K1 construct, we did see some repression. Now it's not at the levels that we would like to see, but it's kind of at least a very clear indicator and pattern that's maybe moving in the right direction. So we're following up on that. And then lastly here, we did see that the selumetinib had some expression in the liver and actually repressed 2 other downstream genes, but we did not see that with any of our nanoparticles. So that was an interesting finding as well. Just kind of another piece of the puzzle that we're working to put together. So in conclusion, we have a number of plasmid constructs that we're able to sustain our repression over 3 weeks. We've identified some clear patterns in our polymer nanoparticle design parameters, that these enhanced our performance, manufacturability, and transfection efficiency for Schwann cells. We've had comparable characterization performance results, even with our scaled up and scaled out manufacturing products. These PNPs were well tolerated. We did see some reduction of MAP2K1 in the female sciatic nerves, and there's more to follow up on with the selumetinib that we observed in the liver. But right now we are looking to improve upon using some different promoters, and also investigating some of the fundamental mechanisms, cell uptake and release mechanisms, to improve our performance. So in general, to close, our HitScan platform enables a lot of capabilities, payload capacity. So this is a large payload, manufacturability, lower toxicity, and payload diversity. And I want to thank the Battelle non-viral gene delivery team. It takes a village here to perform all of this work. So this is a really fantastic team to work with. I also want to thank my collaborators and our funding sources, and I'm happy to take any questions.
Thanks. Great talk, Ken. That was interesting regarding the kind of the sex dependent repression that you saw. Is that you were measuring repression? You were like it was a functional assay.
Yeah, it's I can get back to that.
Yeah. I was just wondering, yeah, so correct me if I'm wrong. It's a functional assay. Do you have any way to measure like biodistribution of the particles as well? Like to see if it's actually like an uptake thing?
That's where we started with our initial screening. We were looking at biodistribution and found designs that did go in large part to the brain and the sciatic nerve. There's a number of rankings of those designs where some only went to the sciatic nerve, some only went to the brain. We question, is that real? Is that not real? But we kind of iterated off of those designs. We used those in this study, but we are able to track that using our barcoding technology or other fluorescent markers. We did not see a difference in sex on that. So yeah, that's a good question.
Hey, great talk. My name's Jacob White. I'm from Loma Linda University. And I was wondering, what are some advantages or disadvantages of these nanoparticles versus something like exosomes or liposomes?
Yeah, that's a great question. So a couple things. So one, immunogenicity. So these are very inert polymer products. Stability. So they can sit on the shelf for a long time. Manufacturability, as you saw, is kind of a key parameter. I know LNPs are much farther ahead in the translated race right now, but we do have a number of advantages there. And also from a exosome standpoint, maybe the immunogenicity might play a factor there.